Hello and welcome to today's webinar. To allow everyone to get connected to the platform, we'll have a brief pause and begin the webinar shortly. Thank you for joining us today. The What's New in APX webinar will start shortly. We're allowing people to have a few minutes to connect to the platform, and we'll begin in just a moment. Welcome to today's webinar, What's New in ADMET Predictor APX, Multi-Threaded Property Prediction, New Transporter Models, and AI-Driven Drug Discovery, and more. A few housekeeping notes. For optimal video and audio connection, we recommend you close out any additional web or streaming applications that may affect your internet bandwidth. We take your privacy rights seriously, and by attending this event or participating in the Q&A session, you're allowing us to contact you for future follow-up. This webinar is being recorded and will be available on our website and YouTube channel. For the Q&A session, you may ask questions at any time via the questions panel on your dashboard. You can submit questions there, and we strive to have at least 10 minutes of Q&A, so please don't be shy. If you need assistance throughout the webinar, raise your hand and our support staff will assist you. We know there are lots of ways to spend your time, and on behalf of everyone on the APX team, we appreciate you spending the next 60 minutes with us. Now to get started and tell us a little bit about yourselves, are you currently using ADMET Predictor software? We'll give you a few moments here to answer. Thank you, everyone, for submitting. And it looks like we have a fair share of people new to ADMET Predictor or haven't started using it yet. So now it is my pleasure to turn this over to introduce Eric Jamois, Business Development Director. Eric, take it away.
Well, thank you, Arlene, and hello, everyone. My name is Eric Jamois, Director of Business Development at Simulations Plus. On behalf of Simulations Plus, I welcome you to today's webinar, which will discuss and demonstrate the new features in ADMET Predictor 10.0, termed APX. It contains new modules and improvements to the software, which you will see. We have a lot of information to share with you. So without further ado, I am introducing David Miller, our VP of ADMET Cheminformatics. And joining David as panelists are Michael Lawless, Senior Principal Scientist, and Bob Clark, Senior Research Fellow. David, the floor is yours.
Okay, thank you, Eric. And again, thank you all for attending today's release webinar for ADMET Predictor 10, or APX. The software was just released and is now available on our ShareFile site. So today I'm going to start with a very brief introduction to ADMET Predictor as a whole. And then I will give an outline of the major new features that we've added in version 10. And then finally, in the remaining time, I will give a demonstration of some of those new features. Okay, so AP overview. ADMET Predictor has four main pillars, if you will. The first and the most well-known is property prediction. So as of this version, there are more than 175 predicted properties, and they are divided into these categories. Physicochemical includes pKa, log P, solubilities, permeabilities, and so on. Metabolism focuses primarily on cytochrome P450 enzymes, but also covers UGTs and aldehyde oxidase. There are enzyme-specific substrate, non-substrate, and inhibitor, non-inhibitor models, as well as kinetic models for the 5 major CYPs and models of overall intrinsic clearance in liver microsomes and, as of this version, hepatocytes. There is a new transporters module that I'll discuss separately. Toxicity includes a variety of diverse endpoints, including Ames mutagenicity, cardiac toxicity involving hERG, carcinogenicity, and many others. And finally, we provide a number of risk scores that predict overall ADMET liability in the form of a single number. The pharmacokinetic simulations are provided by our HTPK, or high throughput pharmacokinetics module. That allows estimation of fraction absorbed and bioavailable and a number of PK parameters. You can estimate concentration time profiles, as well as the dose required to achieve a target plasma concentration at steady state. We have a model building engine in our ADMET Modeler platform. This is the engine that we use to build our commercial models, and customers can use it to build models using their own data, whether those be activity models or ADMET or other property models. The package includes our proprietary set of over 340 molecular and atomic descriptors. It supports both regression and classification models, and regression model predictions come with an uncertainty estimate, and classification model predictions with a confidence estimate. And then finally, we have a cheminformatics platform with a number of tools related to compound design, clustering on the basis of common scaffolds, R group analysis, tools for analyzing similarity, diversity, and several others. For this version, we've introduced a powerful new compound design feature that I'll spend some time discussing later. Okay, the vast majority of our property predictions are based on QSAR models developed using training data that we acquire through collaborations from commercial databases or literature. These models are built using our molecular and atomic descriptors. Some of those more advanced atomic descriptors were developed under an NIH grant They are based on a novel method of estimating quantum level partial atomic charges using a fast empirical approach. And the quality of those estimates is illustrated here. We use these and our other descriptors to build a collection or ensemble of neural network models where the final predictions are the average of the individual predictions within the ensemble. This graph shows the performance predicted versus observed of our PKA model, which is one of several that have ranked at or near the top in external challenges and third-party validation studies. You can consult our website for details on those, and the user manual has similar plots for all of our models, along with a description of the data that we use to build them and notes on our curation efforts, which are usually extensive. Okay, that was the briefest of brief overviews of Predictor as a whole. I had some slides on the pharmacokinetic simulations, but was reminded that we did give a full webinar on the HTPK module, and that is available for playback on our website. So I am going to move on and talk about some of the significant new features that we've added for version 10. One of the biggest ones is a new module that we call AIDD, which stands for Artificial Intelligence-Driven Drug Design. So this provides capabilities for what we call evolutionary multi-objective compound optimization. To explain the AIDD methodology, it helps to refer to a feature that we've had in ADMET Predictor for some time, which I'll call analog generation using rules. So here, a starting structure is modified using a user-adjustable database of transform rules. Those rules can include things like bioisosteric replacements, actual synthetic reactions, or just mutations like change carbon to nitrogen. But chemically intelligent ones in the sense that we avoid creating analogs that would be unstable, non-drug-like, or otherwise undesirable. These rules are applied combinatorially to generate a potentially large library of analogs that can provide chemists with new ideas for synthesis. So AIDD builds on this transform-based analog generation approach with improved and expanded rules. So we spent a great deal of time adding new rules and modifying the existing ones to be more structurally precise and specific. AIDD incorporates property prediction, and those can include our ADMET properties, fraction absorbed or bioavailable, various risk scores, and any activity models that the user has built with our ADMET modeler package. It also incorporates our estimates of synthetic difficulty, which I'll briefly describe on the next slide. And then, unlike the existing feature in ADMET Predictor, AIDD uses an iterative approach. So in each generation of potentially multiple generations, the best analogs are retained using a Pareto optimization approach. Okay, just one brief background slide here. Our synthetic difficulty scores are based on a fragment dictionary approach similar to one developed by Peter Ertl and coworkers. The scores reasonably predict the synthetic difficulties reported by chemists, and that's shown in the graphic here, and the details are provided in this article. This graphic illustrates the concept of Pareto optimization using layers. So here there are two properties or objectives, as we call them. Lower values are better for both of them. So the best compounds are here in the lower left. A first Pareto layer consists of compounds for which no other compound is better in both dimensions or both objectives. So that's shown here in orange. A second Pareto layer can be selected by removing the compounds in the first layer and then repeating the analysis and so forth. So there are 3 layers shown here with different colors. So using this layer approach, you can essentially select any number of Pareto optimal compounds. So the overall AIDD workflow is as follows. You initialize with 1 or more seed compounds and choose a set of objectives for optimization. New compounds are generated by transforming randomly selected compounds from the current population, which initially is just the seeds. Then objectives are evaluated. And again, that can include ADMET, the HTPK, risks and user activity models. And then the population is pruned by retaining only those compounds that are Pareto optimal. Then that process is repeated for some user adjustable number of generations. Population is expanded with the transforms, properties are evaluated, population is pruned with the Pareto optimization approach. So in that way, you can rapidly generate virtual compounds that are simultaneously optimized against multiple target objectives. We have experimented with this for many generations, more than 1,000, and observed that up to about 10 million analogs can be evaluated in a day using a typical machine with 4 physical cores. And I will give a brief demo of this after the slides. As I mentioned, APX introduces a brand new module focused on transporters of particular clinical significance, all explicitly referenced in recent guidance documents from the FDA, shown here, and the EMA in Europe. Some well-known examples of transporter-mediated drug-drug interactions, or DDIs, is shown in this graphic, which comes from the FDA website. And then just for reference, this illustrates that most transporters relevant to DDIs are expressed at the epithelial barriers of intestinal enterocytes, hepatocytes, and proximal tubule cells in the kidney. So with 18 brand new models, this module covers nearly the complete matrix of substrate, inhibitor, and KM predictions for the 9 important transporters shown here. I will note that obtaining KM data for the efflux transporters is difficult due to the relevant substrate concentrations being intracellular ones. Similarly, we have not been able to build Vmax models. And here the issue boils down to not knowing the transporter expression levels within the assay systems being used. We will continue to investigate the possibility of building those missing models, as well as models for new transporters. And as I mentioned, the user manual has the complete details on the performance of these models, the data sources, and so on. We made a number of enhancements to our HTPK simulation module, mostly driven by an ongoing collaboration with a large pharmaceutical partner. Among the new features are support for longer and multiple dosing intervals. We've added several new command line options, which I will try to demo. We have additional PK parameters like half-life and clearance, and the collaboration is ongoing. So we expect further enhancements in the coming year. The graphics here show multiple dose intervals, an interval longer than the default 24 hours. And this is a alternate front end, so not ADMET Predictor, but populated using the output from the command line version of ADMET Predictor. So we are now assisting a growing number of customers with developing these kinds of custom front ends. We have added 2 new models for hepatocyte clearance in human and rat. And we have improved some of our existing models through the addition of new data. And that includes volume of distribution, the blood-brain barrier classification model, the HERG classification, as well as PIC50 model, and then 3 transporter models that we had previously in 9.5, OATP1B1 inhibitor and PGP substrate and inhibitor. We have expanded the parallelization capabilities of Predictor. Now both ADMET property predictions and PK simulations can take advantage of multi-core CPUs. We found that a roughly 4 to 5-fold speed improvement can be expected on a typical machine with 4 physical cores. An even greater speed improvement can be expected for PK simulations, and that's due to many enhancements to the equation solver. This parallelization is enabled both in the graphical interface and the command line, and it does not require an additional license. These are some smaller features that nonetheless were specifically requested by some of our users. One is greater control over the text size in the various display windows. So this is useful when you are giving a presentation or creating graphics for a report. We've enhanced the feature to filter the main spreadsheet using specified ranges for selected properties. Tooltips for predicted properties can now be exported. This is in particular for the ADMET and the HTPK properties. So you can export those even to Microsoft Excel. You see the second row here with the tooltips. That's an optional feature. There is support for heteroatom stereocenters, like the sulfur shown here, and for hydrogen isotopes. You can export compounds using a version 3000 SD format. There are some improvements to license handling. So there's a greater attempt now to preserve customer licenses. And there's also a feature to see who is using a particular license feature at a given time. And there are many, many other user-requested enhancements. We provide a list of all of these in the README file. And of course, the user manual has all the full details. Okay, so with that, I will soon start the software demonstration, but I believe that Arlene has another poll to provide first.
Yes, thank you. Those enhancements are exciting. And as a reminder, current licensed users to ADMET Predictor can download the latest APX version that David has explained. Now, we'd like to know what your primary interests are in joining us today, and feel free to choose all that apply. We'll go ahead and take your answers, and David will start preparing for the software demonstration shortly. You can also keep your questions coming in by using the questions panel. Great. So it looks like a lot of you are here for the enhancements to ADMET Predictor. And without further ado, David, I'll give it right back to you.
Okay, thank you very much, Arlene. Okay, so now I am going to launch the new APX, hot off the presses. Okay, much the same look and feel. I'm going to start by opening up a data set. This is an SD file containing 5,000 compounds from the Kimball database. So I will open that up. To generate ADMET properties, it's the same workflow as before. I apologize if the screen is a little bit hard to see here, but you'll notice there's a new option for transporters now. Now, before I calculate properties, to give you a feel for the speed improvement, I want to bring up the prior version of Predictor. This is 9.5. I've loaded the exact same set of 5,000 compounds here, and I'm going to launch the exact same calculation here. So I will start it in 9.5 and then go to APX and start it here. Now I have set APX to use 8 processing threads, which should be available on a typical machine with 4 physical cores. This machine here that I'm using today happens to have 6 cores, so I can easily run the 2 versions of Predictor side by side. So the calculation for this number of compounds is quite fast. Doesn't appear to be quite as fast as it was when I tested it. I'll have to check my settings here. I may have it set to run on fewer than 8 threads. Okay, now when I go back to AP95, you can see that it's only about 20% of the way through that, that calculation. So dramatic speed improvement. Okay, I'm going to close 9.5 and just come back to APX. I disabled most of the star plot displays so that I could show just the 2 new ones, and these are related to transporters. One for substrate predictions, one for inhibitor. So these are, convenient ways to be able to summarize a lot of information in just a small number of columns. So the long wedges would indicate which transporters this compound is predicted to be a substrate for, and then a similar thing for inhibition. You can, of course, add your own star plots as well. There's a new tab at the bottom called Transporters, where you could select just the 24 new models from that module. And then you can click All to go back to the full display. Okay, now I want to spend a little time showing features involving a particular kind of file that we call a query file. These are files containing arbitrary queries, and they have a number of uses within Predictor. I'm going to show a file here. This one we added for APX. It's called drug-like filters, and it contains a number of queries for undesirable fragments. You can see names like thiocarbonyl, alkyl halide, etc., with definitions in the Daylight Smarts format. We added this file in particular for the AIDD module to further help prevent the creation of undesirable analogs. Users can, of course, modify these kinds of files. Now, one of the things we did in APX was to develop a unified format for these query files so that they can be used without modification throughout the software. So I want to briefly just mention 4 of the uses of those files. The first one is to assign scores to the compounds in the spreadsheet. And the scores will be equal to the number of queries in that file that are violated or triggered. So I can use an option called Add Attributes and select the query option and then load that file. So that file, again, it's called drug-like filters. Okay, I forgot to mention that most of these queries in the file are taken from the literature, and there are references provided in the file itself. So when I run that, it uses all the queries, tests all the compounds, and then assigns a score where the score is equal to the number of queries that are triggered. I can show the histogram of scores here and put it on the y-axis here. So you'll see that the scores vary from 0 to 5. A second column is added that provides the name of the individual queries that were violated. So you see the names here. You can display those names as well in the histogram. So you see the individual query names and the number of compounds that hit each one of them. With this new version, you can order those by frequency. So you see the hydrazine query hits the most compounds. You can view the compounds this way, select them in the spreadsheet using a shift click operation. And if there are too many queries to fit on a single page, we've added the feature to navigate through them. So these are the less frequently occurring queries for this data set. I think the capacity here is for something like 1,000 queries. Okay, so that's one feature of these query files, is assigning scores. A second is that as of this version, you can use those files for substructure searching. So if I launch the query tool, normally you would use this to, for example, launch our sketch tool, MetChem Designer, draw a query, and then bring it back to do a search here. But you can also load a query file. It's preloaded here because we just used it in a different feature. So if I say OK, it's going to select all the compounds that hit at least one query from that file. If I show the selection here in the scatter plot, you'll hopefully see that all the compounds with a score greater than 0 are now selected. So that's another use of these files. A third one, I'm going to select a compound that has multiple query hits here. In version 9.5, we added a new feature called structure sensitivity analysis, which is used to kind of see a picture of a molecule and a graphical representation of why we assigned a particular prediction for a particular model to that compound. This compound here has a couple of nitro groups. We do predict Ames mutagenicity for this compound. If I show it in the SSA window, normally you would use this window to select a model. Like I can select one of our Ames models, and it would highlight a region that is associated with our positive Ames prediction. for the compound. But another use of this window is to open one of those query files. So I can select that same drug-like filters file here. And if I just move over here, it will highlight, these same queries that are shown here in the spreadsheet. The first picture is sort of the concatenation of all of them. And then you can navigate to see just the nitrogen sulfur bond, the diketo group, and the nitros. So that's another use of these query files. And then finally, I mentioned that they can be used within our analog generation features like AIDD as an additional means of filtering the analogs. I am going to show that AIDD feature soon, but I want to see if I can squeeze in a quick demo of the command line. So I've just brought up this is a Cygwin window. So obviously I'm on Windows now. I should say, as an aside, that the APX Windows version is out today. We anticipate that the Linux version should be available in just a few weeks. The command line run that I'm about to do, will be possible on Linux as well once that version is out. So here in this folder, I have an SD file. It's a small one. And I think I have the command I want to run here loaded. I apologize again if it's a little hard to read, but I'm going to walk through the parameters here. This is a shortcut to the executable location. Both the input file and the output file will be SD format. There's a new option to specify the number of threads, processing threads, that you want to use for the calculation. I am requesting HTPK properties, so fraction absorbed and bioavailable, as well as the PK parameters, and both the human and rat predictions will be returned. There's a new option to export concentration time profile information, which goes in a separate file. There's a new option here where instead of using just the single default parameter file that we distribute, you can now create multiple such files, like one per project, and specify which one you want to use. An example of using this kind of file would be if you have, let's say, experimental solubilities, and you want the simulation to use those instead of predicting solubility, this file would contain the mapping, so to speak. So which field from the SD file has those experimental solubilities? And there are many other parameters that you can specify here. And then finally, this is the SD file. So if I run that, it should be very fast. It's 5 compounds. I just want to show as the output, you get both an SD file, which is going to have your PK predictions in it, along with the structures, and then there's a separate file for the CP time information. And I'm going to try just in a minute, if I can, just to kind of show what that looks like. I'm not an expert Microsoft Excel user here, but I'm going to see if I can find that file. There it is. I'm going to load it. I'm going to just quickly filter it so that I can show just one compound out of the 5, and then hide the rat so we can focus just on the human predictions. And then now I can select the time and concentration columns, and I should be able to add a chart that shows the CP time information. I should have mentioned that in the parameter file, I specified multiple dosing intervals. So you see this kind of curve here. So this is relevant to the slide I showed with these alternate front ends. So we have a customer that has a very slick interface where a chemist, can enter a compound, and get back not only predicted properties, but even graphics like this with PK parameters. Okay, back to AP. I have just enough time for a quick demo of the AIDD module. Now, I mentioned that 2 weeks from today, we will have a separate AIDD webinar. The demo there will be more comprehensive. It will incorporate a biological activity model built with ADMET Modeler. For the short demo today, I'm going to focus on the simpler task of improving only ADMET properties. Just briefly, I want to show one slide here. I recently came across this article from a group at Uppsala University. The paper describes structural changes to ciprofloxacin, which is a BCS class 4 drug. The goal was to increase solubility and permeability, in particular by converting the acid group to a simple ester. So I will be using the AIDD tool to see what kinds of esters we can discover by optimizing our predicted permeability and solubility. I want to say that even though this example is a little artificial, there's no consideration of activity at all. We do know that there can be cases where one region of a chemical structure is mainly responsible for activity, and some different region can often be modified in order to improve ADMET. So this is still an interesting case study, and it will give you an idea of how the AIDD tool can be used. Okay, so if I go back to AP, I just so happen to have ciprofloxacin here as the first compound. It is in the Chembl database. We do predict both low permeability and low solubility for this compound. I can show we have a tool to look at the BCS class assignment. So you can see that although somewhat borderline, it is categorized as BCS class 4. And this is at 3 typical doses of 10, 50, and 250 milligrams. Just as an aside, our ADMET risk flags the compound for low permeability. You see the PEF here. The predicted solubility is just above our risk threshold, so it's not flagged for solubility. The mute code that you see here indicates Ames mutagenicity. And it's interesting, this compound and other fluoroquinolone antibiotics do test Ames positive in a particular strain, and we do get that prediction correct. Okay, so if I select the compound and launch the AIDD tool, this is the first interface that you see. I should just say, we are going to go through this more slowly and in depth in 2 weeks. I'm going to select 3 objectives here. The first is synthetic difficulty, which I described briefly in the slides. And this is mainly to reduce the complexity of the generated analogs. I'll select our intestinal permeability model. And I will select our model of solubility at a specific pH. The paper specifically mentioned a pH of 6.5 for the experimental results, so I can enter that here. So I have 3 objectives. The next sort of wizard page allows you to provide some filtering criteria, one of which is a scaffold that all of the generated analogs will have to contain. So here we want to limit the analogs to esters. There's a way to do that. We've added a feature in Designer, our sketch tool. So I can launch that, and essentially what we want to do is, preserve this structure, but also prevent, substitutions from most of these positions. We want only substitutions from this oxygen to create esters. So if I right-click the structure and say copy as query, there's an option here to limit the substitutions at all the atoms. So if I say OK and then paste it, I'll blow it up. These sort of annotations here indicate no substitutions at these positions. So I can just turn that off for this one atom and then select the scaffold and bring it back. Okay. By default, we do select that drug-like filters file that I discussed a few minutes ago. That can, of course, you can launch it here, you can make changes to it, etc. There's one final wizard page with a lot of other options, including, the database of transform rules. You can turn those rules on and off if you want. We will discuss all of this thoroughly at the next webinar. For today, what I want to do is just set the number of generations to a small number. I'm going to set it to 2, just so that I can run it in about 60 seconds. So now I'll click run. So just as a recap, what it's doing is taking the original structure and making lots of analogs of it using this internal database of transformation rules. It is then predicting properties for all of those analogs. And we specifically selected the synthetic difficulty, intestinal permeability, and solubility at pH 6.5. Then it prunes back that population using the Pareto approach. And then that resulting population is sort of fed back into the process. The population is grown, properties are predicted, and then the population is pruned. Once it's all done with the final population, there is a final round of ADMET property predictions so that you can be presented by that at the end. Okay, so it's on the 2nd generation now. It's generating ADMET properties for the final population. And then those resulting analogs are going to be shown in a new ADMET Predictor window. Okay, so here's the list of analogs. There's something like 550. The user can set that parameter. How big a population do you want? I'm going to focus on this chart here. This is showing predicted solubility and permeability. It looks strange. There's some outliers here. These actually have a negative predicted permeability. And that's because these compounds are what we call out of scope. So these, I think, 3 or 4 compounds are outside the applicability domain of our permeability model. And for that reason, they're penalized to reduce the likelihood that they will end up in the final population. That's a feature that you can turn off, but it is enabled by default. I'm going to convert this to a log-log plot, and so you won't see those compounds. So here's the distribution. We have added a feature where in the spreadsheet, you can do a Pareto optimal selection. So I'm going to quickly run that, specifying our permeability and solubility. And then if I show that selection in the graph, you can see the highlighted compounds are right on the front or the boundary. So these are the most optimal compounds from a solubility and permeability standpoint. I'll just hide the rest. So we're left with these. And if I focus on this compound kind of in the sweet spot here, This is about the simplest analog that you could envision for this little experiment here, this methyl ester. And the paper that I referenced, they only considered a handful of analogs, but this was the best one from their point of view. They experimentally measured both permeability and solubility. This did become a BCS class 1 compound. And I can show that too in the BCS class window, that it shifted, still somewhat borderline, but clearly class 1 now. The increase in permeability, is to be expected, given that we've removed an ionizable group. The increase in solubility, which is dramatic, is a little more surprising. But the last thing I'll just quickly show is if we send this over to our MedChem Designer software, I'll just quickly put both compounds here. So that's the original with the acid, and that's the ester. If you look at the predicted solubility profile, so versus pH, you see that there's a minimum in the physiological pH region because the compound is a zwitterion. So there's positive charge and negative charge. And contrast that with the ester, where now there's just the cation group. So in the solubility profile, in the physiological region, the compound is ionized. So there's a much higher predicted solubility there. Okay, looking at the time, that was a very quick demo of the AIDD feature, but I hope it gave you a sense of how it can be used to discover some interesting and chemically reasonable analogs that also have a good predicted property profile. And with that, I'm going to thank you all again for attending. Remind you one more time about the webinar 2 weeks from today, same time, that again will focus on just the AIDD module. So thank you again, and I'm going to pass it back over to Eric and Arlene.
Great. Thank you, David. This was an excellent demonstration. Now, before we begin the Q&A, just a final quick question. Based on what you've seen and heard today, would you like us to contact you so you can begin your evaluation of APX? We'll give you just a few moments to answer that while we get ready to turn it over to Michael and all of the questions that have been coming in so far. Great. Thank you. Okay, Michael, you are on, and let's get started with these questions.
Okay, thank you, Arlene. Okay, so the first question is for users of GastroPlus that have the ADMET Predictor module, what enhancements have been made for the module, and how might they help PBPK scientists to better understand their molecules?
Okay, maybe I'll start, and you guys can think if there's anything else. I think our plan is to release the next version of GastroPlus in, I think by the end of this month, but if not, shortly thereafter. And I know one thing that should be available are the transporter model predictions. I believe that those should be brought back into GastroPlus. And of course, the models that we've improved, like for example, the volume of distribution model, that newer prediction will of course be available in GastroPlus as well.
Okay, thank you. I think that should answer the question. And then the next one, David, is about the star plots. So if you can bring back up ADMET Predictor, and the question is, what do the colors signify in the star plots? Why are some hatched and some others are solid?
Okay. The color scheme, I believe, is somewhat arbitrary, but there's a tool here where you can you can sort of customize it to your liking. And you can do other things here, like put something on a log scale. You can put a minimum and maximum here. The sort of hatched wedge here indicates that this particular prediction is out of scope. So if I were to scroll over to this PGP substrate model as a number within the spreadsheet, it would be underlined and colored red. So that's just sort of a visual clue that that's perhaps a less reliable prediction.
Excellent. Thank you, David. Another question is that this person's dealing with molecular weight compounds above 900 Daltons and wanted to know, what is the molecular weight cutoff for an accurate prediction?
Perhaps Bob will want to chime in here. I can just sort of from experience say that, again, our models are built on training data. There's certainly an underrepresentation of compounds of that size in our training sets. So I think in general, the models, most of them would probably be, somewhat less accurate. You will get an indication of that. We just talked about this out of scope prediction. So you would get some sort of visual clue as to whether or not the compound predictions were in scope. I'll see if Bob has anything to add to that.
Yeah, it depends a lot on which model it is. The out of scope gives you a heads up. We've actually checked them out, many of the more commonly used ones for things like permeability and solubility, and they're actually surprisingly good up to about 1,000 molecular weight. What's called millimolecules in the trade. The other thing is, because we have the uncertainty estimates, that gives you another handle on whether you can expect them, on how reliable you can expect them to be. But there are more representative in more of the training sets than you would think. And because we rely on descriptors other than just substructure descriptors. Many of the descriptors we use are whole molecular property descriptors. They're a lot more reliable. They're a lot less size dependent than most of the other ADMET models on the market.
Very good, thank you. So then another question is, just repeat the fundamental theory that is used for the prediction. So just quickly talk about the models, how they're created.
I didn't catch that, Michael.
Repeat the fundamental theory that is used for the prediction. So how are the models created?
Yeah, so in just a brief nutshell, so they're based on training data. We of course, split the data into the training portion and the external testing portion. I didn't mention that earlier, but we start with our proprietary molecular and atomic descriptors. We build models, which tend to be artificial neural network models. We don't build just one, there's an ensemble of them. We vary the complexity of the models, fewer and larger numbers of neurons, for example, and then have a sophisticated way of choosing the best model, that incorporates performance on both the training set and the completely external test set. That's the overall process for model building in a nutshell, and I'm sure we've given one or more webinars on it that are probably available on the website.
Yes, okay. Let's see, how do you get a license for APX? Maybe Eric can chime in with that.
Yeah, so each module is licensed separately. By default, the evaluations would not necessarily include the AIDD. If you are interested specifically in the AIDD aspect, please mention it to us, and then we'll make sure that you get that license feature as well. But otherwise, evaluations come with all features activated. So if you're interested in pursuing an evaluation, please contact me directly. And I'll be happy to work with you towards that. If you're a nonprofit or academic organization, you'll be working with my colleague, Cheryl-Ann Yagala.
Thank you, Eric. Yeah, if you could go back to the AIDD example and show a few more of the esters on the Pareto front. Someone would be interested in just looking at those. So go back, I guess go back to, if you can bring in the input, the output file from AIDD. And then just maybe, you know, put your graph and log units and show a few more of the compounds there.
We could say a lot about, you know, the AIDD tool here. It's very flexible. You know, I showed just a fraction of the options that are available. Typically in kind of a real life, you know, drug discovery, you know, scenario, you would be spending quite a bit of time on your filtering rules, you know, kind of limiting what it is that you want to see in the end. I hope that we cover most of that in the separate webinar that we'll have in 2 weeks. This was basically using, you know, purely default parameters. So surely there are, you know, some compounds here that, you know, depending on who you are, you know, may consider to be undesirable. But it's extremely configurable. Let's put it that way.
Very good. Okay. And then I think the last question we have so far is what type of test train split? Random, scaffold based? So how are the training and test sets defined when you create a model?
Bob, do you want to cover that one? Or should I? I can do it.
Usually we use stratified sampling for the external test set. And then for the internal splits, for when we build the ensemble, each split for the each different submodel gets about 2/3 of the training pool, and the other third is held for what we call a verification set. And that is done by a random shuffle. But the external test sets ordinarily will do with what's called stratified sampling, where it's random within each random, but it's spread evenly across the response range of the dependent variable, usually. There's no practical way to do time splits because this is for literature data, if that's what the question gets to.
Okay. Can you go back to the esters? Just show a few more. The person that asked the question said he didn't see the ester structures, and he wants to see them for the Pareto front only. So maybe do the. Can you generate the Pareto front again? Okay. And then maybe on display, or yeah, just there you go. Okay. And then maybe go into tile mode or something. Okay. So those are the esters on the Pareto front. And I think everything's going to be an ester because you left the oxygen there and it's only adding groups to the oxygen there. Okay, yep, that looks good. He's what he wanted to see now. So that's all the questions we have. I'll turn it back over to David, I guess, for some final remarks.
I think it might be over to Eric or Arlene for some final remarks.
Great, thanks guys. You can find the HTPK webinar, Uppsala publication, and request for evaluation form on our website, www.simulations-plus.com. As David said, we look forward to seeing all of you on September 30th, when Dr. Marv Waldman delivers on the promise of AI-driven drug design with the new APX AIDD module. This concludes our webinar for today, and playback will be available on our website soon. Thank you. Have a great day.